Congratulations, you guys made it through Unit 2. We haven't started grading the prelims yet. I still have some outstanding, so we're going to start on those probably later this week. So hopefully we'll have them into you guys before Thanksgiving, though maybe you guys don't want them before Thanksgiving. We're going to start a new section now, okay? And this Unit 3 is really all about management. I'm going to start talking about management by talking about nutrients. Now, the lecture, I mean, you've seen some of these slides. We're going to be talking about nutrient cycling, just sort of to renew you. But actually, you know what? Jean-Marc, can you kill the first bank of lights? I forgot to do that. I apologize. You guys can see that? Cool beans? All right. And common conversation, it's essential for our life. All its availability or lack thereof is really controlled by it really controls the distribution of flora and fauna. And in many cases, the distribution of flora and fauna also controls the nutrient cycling. Certainly the nitrogen cycling is a good example of that. Okay? And when we talk about nutrient cycling, we're really talking about these three factors. What's controlling the pools, both the sources and the sinks, and what's the control between them, the fluxes. Okay? So, this is cutting off the screen. We've lost half the, or a chunk of the slide. I guess it's okay. And again, you guys have seen this slide as well. The larger sinks of nutrients are generally unavailable to organisms, but there is always a group of organisms that are involved, with the exception of potassium, but potassium is usually not a limiting nutrient, that specialize in operating on this unavailable pool, making it available to everything else. Okay? Much of this transformation, and this is why you're in a soils course, this is why I'm talking about it, much of this transformation is soil related and is controlled by soil characteristics or conditions. So what we're going to do for the next 3 lectures is basically follow this flow chart. We're going to be talking about, again, nutrient cycling, but from a different perspective. We're going to be talking about the management of those nutrients. We're going to be talking about nutrient management specific as well as fertilizers. And then finally, we're going to be talking about environmental dynamics and the quality of the dynamics on those nutrients. Or not just on nutrients, but on other management issues with soils. Okay? Now, the model system that we're going to be operating on is mostly agricultural fields or agricultural crops, whether they're field crops or truck farm type of crops. But keep this in mind when we're talking about forested systems, agricultural systems, as well as grass systems. Okay? We're talking about management of these materials across a lot of different type of plant systems. All right. Now, what we're going to do today is we're going to be talking about nutrient cycling. And when we talk about nutrient cycling and when we talk about nutrient management and fertilizers, which is going to be the next lecture, I want you to keep in mind these 6 things. Now, we've talked about all these. I mean, this is all Unit 1 type of stuff, and some Unit 2. But the first thing I want you guys to think about is the parent material. The texture of this material and the parent material. How is that going to be affecting nutrient dynamics? Okay? Certainly, when you start talking about texture and mineralogy, you're going to start talking about what's in the material as it degrades or weathers and becomes available for nutrients. But also, when we start talking about texture, we also should be talking about surface area. Because remember, the surface area is the edge effect. Everything's going to be happening on that. Okay? Now, the nature between the mineralogy and the texture, you're going to be starting to talk about cation exchange capacity as well as AEC. Okay? And that has to do with that surface and the mineralogy combined. Okay? Also the climate. Climate, for a number of reasons we're going to be talking about. First, climate is going to affect biological activity. But second, climate is also going to affect reaction rates, chemical reaction rates, the C10 rules. And the presence of water allows different reactions or the frequency or not the frequency, the speed of those reactions. Okay? Biology. Obviously, biology has a lot to do with the cycling itself, but also the organic matter as one of the pools. Okay? pH, another really big important one. We just got done talking about pH. pH and its effect on the active acidity pool and its reaction rates with exchange capacity, relationships with soils, but also pH with its effect on solubility of different materials. And then finally, the nutrients themselves. So, The model system we're going to be talking about is an agricultural system, primarily plants. So let's talk a little bit about plants. Plants are made up of basically 75% water, 25% dry matter. Okay? That dry matter is mostly CHO. You guys have seen my CH2O slide all the time, basically representing organic matter. This is the reason why we use that. Okay? The nutrients, we call them essential nutrients. Essential nutrients basically are those elements that are necessary for the plant to complete its life cycle. That doesn't just mean grow, but it also means to make seeds or reproduce. Each essential nutrient, and this is why they're essential, has at least one function in that plant, or in us for that matter, for which no other nutrient can substitute. That is why it's called an essential nutrient. When I was in grade school, I learned this as C. Hopkins Cafe. Those were basically the macronutrients versus micronutrients. There are macronutrients versus micronutrients. Macronutrients mean basically there's an abundance. I learned it as C. Hopkins Cafe. That was the mnemonic that we used. But you can't use Fe anymore. Fe is not considered a macronutrient, and Mg is now considered a micronutrient. Whatever. But the important note here is that there are some of these nutrients that we need a lot of it, and some of them we need very little bit, a trace amount of it. Okay? Some plant species also need sodium and silica. Silica is a big one for grassy type of materials, grass plants, because they need it actually for their structure to keep them upright. Okay? And then there are also the legumes, which basically those organisms that are doing the symbiotic nitrogen fixation. Those guys need to have cobalt. Okay? But the rest of us don't need that, and most plants don't need that as well. All right. Now, I talked earlier in this semester about the barrel analogy, and I want to use this analogy one more time to sort of get us into the flow of what this lecture is about. Okay? You guys remember what the barrel analogy is? Okay? The idea here is that the barrel represents the potential growth for something, and we're going to be talking about plants, so it's the potential growth for plants. Okay? And each stav in the barrel represents a supply of something that plant needs to reach its optimum growth. Now, the reality is the plant will never get larger than the barrel. That's its optimum potential. But it certainly can grow smaller than that barrel based upon limitations on its growth. In this scenario right here, we're looking at phosphorus. You can see the phosphorus is the lowest slat in the barrel. The water is limited to that height because of that slat, and so phosphorus is the limiting nutrient. Okay, if we add phosphorus, fertilizer or something like that, phosphorus, the barrel slat goes up, and the next most limiting nutrient comes into place, and in this case it's nitrogen. Okay, now in this slide we're basically just talking about nitrogen, phosphorus, but if you look at all the slats in here, you have light, you have heat, you have mechanical support, organic matter, phosphorus, nitrogen, and potassium, other nutrients, water and air. These limiting characteristics are diverse. It's not just nutrients. And I put this in here to get us thinking about this issue of management. What are we in fact managing for? Now we're talking about soil management, but whatever our goal is, in essence, is basically this barrel of water. We want this barrel of water to be a certain size. Okay? And we have a maximum size it can be, but we have limitations on that size. The management is basically taking care of these slats. Okay? Questions so far? We good for? All right. This is an experiment that Murray McBride and myself did. Okay? And this slide really, this is actually a toxicity issue. But I want to use this slide, and I'll talk about the toxicity issue in a sense, but I want to use this slide to sort of represent this. Okay? Now, what you're looking at is you're looking at lettuce. Okay? And lettuce is growing in the same type of soil. Okay? Every one of these lines represents a different treatment. Now, can you guys see the treatment line that's got some sort of effect to it? It's definitely, there's a suppressed growth here. Okay? Now, if we think about that in the sense of a barrel analogy, that suppressed growth could be one pot basically having less phosphorus in it. Okay? Everybody else has more phosphorus. Okay? And so we get this type of response. Now, in this case, it wasn't a phosphorus response. In this case, it was actually a pH response. Now, generally, when we think about management, and certainly when we start thinking about this barrel analogy, we generally think of some sort of limitation based on there's not enough of something. But this barrel analogy can certainly be flipped over where, in fact, it's not necessarily something that's not enough of, but it could be something that's there's too much of it. Okay, it could, in fact, be a toxic effect. Does that make sense? And that's, in essence, what you're seeing here. Each one of these pots basically has the same amount of material, but each one of these pots has a different pH. And in this case, we're talking about a very low pH. The pH gets low enough, and I start seeing availability of nutrients or toxic materials that are at higher levels than in these other ones. And what you're looking at here is you're looking at aluminum toxicity. Now, this was an experiment about lead. As the pH of a soil goes down, lead becomes more and more soluble. And our experiment was basically to take a look and see what was going on with plant uptake in lead. But we lowered the pH enough that we're looking at different responses. This is not a lead response. This is a response to an abundance of aluminum because of the lowering of the pH. It increased the solubility of the aluminum. The aluminum went into the soil solution. The aluminum then started affecting the plant roots, and you had the toxic effect, and you had the plant suppression. Does that make sense? Okay. Now, segueing this a little bit, let's think about what happens to those plants to other organisms. Now, obviously, we eat plants, or organisms that we eat plants. Okay? It turns out, if we go back to this barrel analogy, these types of plants may be, I mean, this, the amount of water in here is the plant that we're growing, and we're feeding that water, the plant, to organisms. Now, it's important to remember that not all organisms have the same nutrient requirements than everybody else. In fact, some plants have very different nutrient requirements than other plants. So if we're thinking about this from a management and use, it turns out that non-deficient plants, plants that have the optimum resources that they need, can in fact provide deficient diets to us. We have very different requirement nutrient-wise than plants do. So even though the plant is growing at an optimum level, it doesn't mean that it's going to provide the nutrients that we need. We see this especially with things like molybdenum, selenium, cobalt, copper, phosphorus, sodium, the whole list. But in essence, you got to think about what you're growing, what you're managing it for, and where it's going to. Okay? Plants can contain high levels, even toxic concentrations of essential or non-essential elements that could be in fact toxic to them and not toxic to the organisms that are eating them. Could also be non-toxic to the plants, but in turn could be toxic to the organisms that are eating them. Okay? So you got to think about this barrel analogy not just from the performance of the plant but also from, or the performance of whatever you're interested in, but also from the performance of the thing that's going to be using that plant, that barrel. Okay? Does that make sense? Okay. All right, but we're talking about plants. And now let's talk a little bit more about how the plants actually get these nutrients into their system. Okay? Foliar uptake is, unless we're spraying it, it's not a big deal. Not necessarily a big source of nutrients, except unless you're an aquatic plant. Most plants are basically getting their nutrients through root uptake. And basically, you're looking at a cell wall here, and with a cell membrane, okay, inside that. And basically, what the plants need to do is to get those nutrients from out in the soil into the cell. Okay? Now, they have a lot of different ways of doing that. We're going to talk about that. But the membrane basically functions to concentrate these nutrient ions inside that cell versus outside. Okay? Now, how they do that is primarily through respiration. They're using respiration to get energy. They use that energy then to pump and discriminate those ions inside their cells. Okay? Now, the result of this is basically we have an uptake of nutrients that are sensitive to biologically limiting things. Plants, cells in the roots are basically aerobic. They're not photosynthesizing. Okay? They're aerobic. They need to have oxygen to respire so they can do this. If the system gets cold or there is a lack of oxygen, you're going to limit biological activity, and as a result, you're going to limit this nutrient uptake. How these work, we've talked about this earlier with issues of proton pumps, but basically we're looking at, here's the root tip. The plant cells must maintain a charge balance between the inside of the cell and the outside of the cell. Okay? Now the inside of the cell generally has a higher concentration of nutrients, but they still need to maintain this charge balance. So if they are bringing in cations, positively charged ions, they need to counteract that positive charge. And they can do it two ways. One is they can bring in negatively charged ions, anions. Okay? Certainly you can imagine if they had to pump in potassium, they could pump in a nitrate, for example. Potassium is the positive charge, nitrate is the negative charge. Okay? They also can pump things out. If they're bringing in positively charged ions, they can pump out protons to counteract that change of positive. Positive in, positive out. And this is where we get in the term of the proton pumps. Now certainly you can also imagine that depending upon what their sources of materials are, they could potentially be bringing in anions. And nitrate is a really good example. If nitrogen is their primary source of nitrate is their primary source of nitrogen, it's a negatively charged ion. So they're bringing in negatively charged ions into the system. So in essence, they have to either pump out negatively charged things or bring in positively charged things at the same time. Now, why is this important? Think about fertilizing. If we fertilize our plants with ammonium versus nitrate, two N sources, if we're bringing in protons, so think about the rhizosphere, okay? If the plant is bringing in ammonium as its source of nitrogen, it's going to be pumping out positively charged protons. What is that going to be doing to the rhizosphere pH? If I'm increasing the concentration of protons, I'm going to be lowering the pH, right? Now, spin that. What happens if the plant, not me, if the plant is bringing nitrogen in as nitrate, not ammonium? Nitrate is coming in as a negative charge. Now, it could be bringing in positive charges at the same time to maintain that charge balance, or it could be pumping out negative charged ions. What is it going to be doing to the rhizosphere when it's bringing in nitrate as its nutrient source of N? What's going to happen to the pH of the rhizosphere? It's going to go up. So depending upon what we're fertilizing, we in essence are going to be changing or playing with that pH. And what does pH do to the availability of nutrients? Changes them. If I start lowering the pH, I'm going to start making micronutrients more available off of 6.5. If I start raising the pH, I'm going to start making macronutrients more available as opposed to 6.5. Does that make sense? Okay, so how the plants are feeding also has a huge impact on the environment that they're in and the availability of nutrients. Now, we're talking about this, the plant uptake of those nutrients, okay? But it's important to think about this in a sense, you know, if we're interested in maintaining fertility for our plants, we can't just think about the uptake of the plants, of those nutrients. We need to think about what's in the soil solution and how it's interacting with those reserve areas, okay? Now, if that's what we're talking about, we're really talking about release mechanisms. We're talking about release mechanisms of nutrients, or cations or anions, from this reserve area into the soil solution where it can then go up to the plant. Those release mechanisms are basically threefold. There's sort of three controlling issues here. If it's a cation exchange reaction, cation exchange capacity, it's basically charge related on the exchange sites. That exchange can be pretty fast. On the other hand, if it's the nutrients are literally part of organic matter that has to decay before it's released, that is going to be rather slow. And then if we start talking about dissolution of primary minerals, of the rocks themselves, that can in some cases be very fast, but in some cases can be very slow. Okay? All of those are going to have controls on this flux into this nutrient solution, right? Which then means we have to actually think about what's going on here and here. We got plant uptake, we have exchange reactions, Those plants take that stuff up and that gets harvested. Potentially, stuff that's in the solution can be leaching. So what is the only other source for these nutrients to move into the system? Basically, us bringing in amendments. If we're harvesting the nutrients, not just the plant. The result of this is basically twofold. One is the amount of nutrients that are in the soil solution depends upon the history of that site. Okay? The amount of nutrients that are in this pool right here are going to depend on the history of that, the history of that, the history of that, and the history of this. Does that make sense to everybody? Okay. Now, the other interesting thing about this, the other result of this, is that you have to consider that each one of these nutrients are going to behave differently in that soil solution. Some of these are more soluble than others. Some of these are going to be coming in as fertilizers, some of them are not. Some of them are preferentially going to be leached from the system. Some of them are going to be preferentially lost from harvesting. Okay? Because of this history, you have to think about how these nutrients are individually going to behave as well. And this brings us to this idea of the tank model. Okay? We've looked at this before, okay? But just to remind ourselves, basically what we're looking at here is this tank represents all of the nutrients that are in the soil. This little spot right here is basically the nutrients that are in this soil solution. This is basically the reserve, primarily the cation exchange capacity, the stuff that's in the cation exchange capacity. Though it also could be considered, in a sense, the stuff that's in the structure of the organic matter. Okay? Now, as this nutrient pool is dropped down, the fertility of the pool is dropped down because of use, you're going to have an equilibrium between the active pool and this reserve zone, the exchangeable area. Okay? Now, what we would do is we would add fertilizer, and in the process of adding fertilizer, we would raise the nutrient levels in both the active solution as well as on the cation exchange sites, the reserve areas. Does that make sense? Okay. Now, let's think about what that actually means for the plant. Okay? Here, we've brought up the fertilizer all the way up to here. Okay? Optimum fertilizer levels, optimum nutrient levels in the soil. Okay? So here we start. The plant starts sucking nutrients out. What's going to happen to that active pool? It's going to drop down, but then it is going to equilibrate with the reserve area. It's going to drop that pool down a little bit and raise the active pool right back up. Not quite as high as it was in the beginning, but it's going to raise it back up. Okay? Now I keep drawing off of this, and I'm going to keep drawing this down, and I'm going to keep getting equilibration between the two of them. Right? Yes? No? Okay. Yes? Yes. Okay. Now, the question then is, what is the nature of this pool? Because this is really what we're managing. The plants are managing this for us. We're managing this. We're managing the whole system. So let's look at that tank analogy again, but let's look at it from a perspective of two different pools. Okay? Here is a pool with a low CEC. Here's a pool with a high CEC. Both of them, because of their drop in fertility, right now they're both pretty infertile. Does that make sense? Now, if I want to bring their fertility back up, I want to add fertilizer in some form. Now, to do that, because the capacity here is so small and this is so large, the amount of, let's imagine it's phosphorus, the amount of phosphorus that I would have to add as fertilizer to this system, to these two different soils, if I add X amount right here, it is going to raise the amount of phosphorus very much higher in this system than in this system. This line represents the low capacity soil. This line represents the high capacity soil. Does that make sense to everybody? If I am adding the same amount of fertilizer, here, no, stop. Let's turn it around. If I have the same amount equilibration rate in both soils, okay, starting, let's actually go back one more. Okay, I start at a high fertility soil, right? I put the same kind of plant in. They both have equal amount of demand. Now, if I have two soils, one soil with a large pool and one soil with a low pool, and they both start at the same amount of nutrient availability. They're the same level. They're all at this level, right? All the way across the board. If I put the same kind of plant in both of these, what's going to happen to the nutrient pools? In both of them, it's going to drop, right? It's initially going to drop down here, down to here, down to here, okay? But to make this back up, because there's going to be an equilibrium, this is going to come back to here. This line is going to drop here. This is going to come back to here. It's only going to drop here. So this guy now has availability to here, but this guy has availability to here. Does that make sense? Now, flip it over, though. Think about these when they're at their lowest point. Actually, I don't have to do that. We got that as a slide. Here they are at their lowest points. Which one of these is going to take more fertilizer to bring it back up to the high level? The high capacity one, because I put in the same amount in each case. Say X amount, 100 pounds. In this one, it's going to bring it up this far. In this one, it's going to bring it up a lot higher. Does that make sense to everybody? Yes? Okay, question.
How quickly does it reach equilibrium, especially compared relative to the rates that plants are taking it up?
Okay, so the question is, how quickly is it going to reach equilibrium? How quickly, if I draw this down, how quickly is it going to recharge in essence? Okay, that level, that recharge is going to be based on these three things. If it's a cation exchange reserve, it's going to be fairly fast. If it's organic matter decay, it's going to be fairly slow. If it's disillusionment, it's going to be slow or fast depending on what's being dissolved. Okay, does that make sense to everybody? So the question was, if we get to this state, or I should say, if we get to this state, how long is it going to take to get to that state? How long is it going to take to get back there? And it's going to depend upon this reserve pool. If it's cation exchange capacity, it's going to be fairly quick. If it's organic matter decay, if this pool here is basically an organic matter, it's going to be slow. Now, a good example of this is Dillman Hill. You guys, at this point, we got the results back from Dillman Hill last week, right? Everybody had really high levels of nutrients. Yet, we still suggested giving fertilizer to those plants. Why? We had high levels of nutrients, yet we still asked. Why?
The high levels of nutrients could be in solid forms of some kind, like organic matter. So adding compost makes nutrients more available, or the nutrients in the compost are more available.
At Dillman, your answer is right. At Dillman, what are they using for fertilizer? They're adding it every year. It's compost, okay? Which means a lot of those nutrients are tied up in that organic matter. And if it's tied up in that organic matter, it's going to be the slow decay. So this return is going to be a lot slower. And that is why we suggest adding a little fertilizer for certain stages of those plants' life. Go.
So in that scenario, though, if you're consistently adding compost, would you theoretically be at a steady state where you would have to add fertilizer because you had compost there?
And the answer to that is yes. So the question is, in Dillman's case, if we're consistently adding organic matter year after year after year, would you still have to, because this stuff is decaying, okay, So supposedly it should, at some time, it's got to become available. In Dillman's case, because they've been adding this, this organic matter year after year after year after year, they don't actually have to follow the recommendations because there is, in fact, a lot of nutrients there that are, in fact, available. They may not be as available to certain stages of the plant's life as they're needed, but this is actually a deficit of the nutrient analysis of the protocols that we do for a recommendation. We're basing our recommendations, we're doing chemical analysis, we're basing our recommendations on sort of history of the site. Part of that history of that site that we don't take into account is this continual amendment of organic matter. Okay? Some of you guys had 17 to 25% organic matter in those beds. That's not from a traditional agricultural process or practice, whether it's a horticultural operation or field crops. Those are levels of organic matter that are usually not found. You're lucky if you find 7, 5, 8, 7%. Okay, you are really, really lucky. And in this case, we got 25% organic matter. That's well beyond what we usually see. The reason being, they add this stuff every year. Okay? Questions? So let's actually talk about those release processes. Okay? The first one, and the one that we've been talking about right now, is organic matter decay. Okay? It's especially important for things like nitrogen and sulfur, though it's sometimes also important for phosphorus. Because of this decomposition, we often see great fluctuations in the concentration of nitrogen in the system. Nitrogen is a feedback mechanism for decomposition. So depending upon how much nitrogen and what the CN ratio of the material is, you're going to have very great fluctuations in the nitrogen release. Okay? Fastest release in warm, moist, aerated soils. Well, why? Those are the ideal conditions for decomposition. Okay? Those are, you know, if you want to see the fastest rates of biological activity, you have moist, warm, aerated soils. So if I have fast rates of biological activity, I'm going to have fast rates of decomposition, which are going to release the nutrients. Make sense? Okay, the next one is cation exchange. This is the CEC exchange. Okay, this is the equilibrium between the soil solution and those cation exchange sites on the colloids. Okay, this is especially important for things like calcium, magnesium, and potassium. It features very little fluctuation from those equilibrium points. I have a rapid equilibrium. Cation exchange, I put something into solution, it's going to exchange on those cation exchange complexes almost right away. So what I put into the soil solution as a fertilizer or water or whatever is going to have a very rapid response. Or what I take out of it through leaching or plant uptake. We also have mineral disillusionment. This is basically weathering. This can be fast as well as slow, but it's especially important for calcium, potassium, and magnesium, especially from those primary minerals like apatite or calcium carbonate, but also from oxides, iron, manganese, and aluminum, as well as sulfur from metal sulfide oxidation. Okay? It's most important in young soils. Why? Think about around here, the landscape. We had glaciation, we have fresh rock material that's broken up into small surface areas, so there's a lot of weathering going on. Okay? Give it a couple thousand years, 1,000, 10, 100,000 years, those weathered products have been weathered out, and we're getting older soils. A lot of the nutrients have been weathered out. You start ending up in these altisoils of the southeast, for example. Okay? Give it a lot of 100,000 years, and then you start getting into the oxasoils. Okay? Specific absorption, desorption. Good example of this we talked about with phosphorus. You guys remember phosphorus and issues of pH? At low pHs, phosphorus bound very well with aluminum and iron to chelate, to basically become unavailable at low pHs. At high pHs, it bound really well with calcium. So the optimum pH range for availability was in that 6.5 range. Okay? This is a good example of that type of specific sorption-desorption type of reaction. Okay? We also have surface chelation. This is often associated with organic material. This is chelation. Remember we were talking about chelation where we're looking at multiple ligand bonds? Okay? This is important from nutrient sense for copper, basically the metals Okay? But this is also really important when we start talking about toxic materials such as lead. Okay? All of these processes are also affected by pH. All right. We got through that? You guys feel comfortable? Any questions about that? We good with the 5 processes that basically control release? Organic matter decay, cation exchange capacity, weathering, sorption, desorption reactions, and then finally chelation. Okay, so at this point we have a fairly good understanding about uptake and release. So what I'd like to do now is talk a little bit about transport. Because if you start talking about how these ions are actually moving to the roots to be taken up, we certainly have an understanding about the release, how it gets into the soil solution, and we certainly have an understanding about once it's in the soil solution, gets to the plants, how it gets taken up. So what I'd like to do now is basically talk about transport for a little while. Okay? And if we're talking about transport, we're basically talking about 2 things. Okay? The first thing we're going to be talking about is basically mass flow. Okay? Mass flow is where the ions are moving with the mass of the water. Okay? They're moving with the flow system. Okay? Moving soil water carries along these solutes. Okay? It's responsible, this is also what's responsible for nutrient leaching. Okay? But this movement of water literally will transport those ions to the roots. Okay? We have an example here. Think about this. The plant is respiring. Okay? That means it's pumping water into the system. Okay? Now that water is going to go out and it's going to go up through transpiration. It's going to go into evapotranspiration, right? It's going to be leaving the plant. What it's doing though is it's creating a flow path of water into the root. Okay? If these ions are in this soil solution, the water as it moves this way for uptake is going to bring those nutrients with it, which gives the plant the availability or the opportunity to take those ions up into the plant. Okay? Does that make sense for plant uptake? But that's how the ions make it from the colloid or wherever it's going into the soil solution and make it to the root. Cool beans? The second one is diffusion. Okay? And this also has a lot to do with the plant. But in this case, when we talk about diffusion, we're talking about the movement of individual molecules or ions across concentration gradients. This is not the water moving it. This is the concentration gradient. If I have a high concentration here and a low concentration here, the concentration in the high zone is going to equilibrate with the low zone, so the ions that are over here are going to be moving over to where the low concentration is. Does that make sense? Yeah? Yes? No? Yes? I am going to have to start having coffee at the side of the room. Really, no, really, does this make sense to you guys? Yeah? Okay. All right. Now, interestingly enough, this is also driven by the plant. Okay? The plant in the first place with mass flow was basically sucking water up. Okay? Well, in this scenario, the plant is also sucking up ions, not water, it's sucking up ions. And if it's sucking up ions, what it's doing is it's creating along this root edge a relatively low concentration of these ions, which means there's going to be a high concentration over here and a low concentration over here, which means there's going to be equilibration. So there's going to be diffusion of those ions into this low zone of concentration over here. Now, this is a slow reaction compared to mass flow, where the water is just moving. But this still does drive some of these nutrients. Now, it's important when you think about the nature of the nutrients. Some of these nutrients are more soluble than others. If I have a very soluble ion, it's going to basically move wherever the water moves. But if I have an ion that's not so soluble, that mass flow is not going to be as important, and diffusion reactions are going to become more important. Does that make sense? So take a look at this next slide. Okay? Optimum transpiration is met when I have optimum mass flow is when I have transpiration. Okay? So the nutrients that are moving with mass flow are going to be optimized to get to the plant when the plant is transpiring. But if I have a nutrient that is not as soluble as the others, it's not going to be moving that quickly with the water. And it's going to be more moving based on the ion transport, the plant uptake. It's going to be moving based on this diffusion gradient. Does that make sense to everybody? Now the reality is it's a combination of both of these, and because of just the mass of movement, mass flow is probably the more important. But you can't disallow this really important role of ion diffusion. Okay? Oh man, is it that bad? Okay, so in essence, that's this slide. Okay, we have mobile nutrients and basically we have immobile nutrients. Mobile nutrients, sufficient nutrient ion basically supplied to the plant by mass flow. Okay, examples here are calcium nitrate. Now, I say nitrate if not deficient. If there isn't any nitrate in the system, mass flow isn't going to move it because it's not there. Okay, so this is a fertility issue. If I have to have nitrate in the system, I have to have nitrate in the system for it to be moved by mass flow. Okay, now, immobile nutrients, the rhizosphere depletion basically makes the diffusion an important issue. Okay? Some of the mechanisms here, we're looking more at the phosphorus, iron, zinc, molybdenum. And this is the interesting thing here. Let me go to the next slide, actually. The interesting thing here is the plants take advantage of this. Okay? If all the nutrients move through mass flow, let me do this as a slide. Don't draw this. Now, if all the nutrients basically were very soluble, why does it make sense for a root system, a plant root system, to become really extensive? Okay, wouldn't it make sense more for me to be a plant to have a well system where what I do is I transpire at really rapid rates? Because what I can do is I can just keep sucking water to me. Does that make sense? If I keep sucking water to me, the nutrients are coming because they're going to be going with the water. I don't have to do a lot. Does that make sense? But the problem here is that I have nutrients that are not necessarily moving with the water. So in a sense, I have to move to them. And I have to create a diffusion gradient. And that's why I have a system that I have lots and lots of roots. Because each one of these root edges becomes a diffusion gradient. Does that make sense? I start pumping ions into the system. I create a low concentration around the root, which encourages these nutrients that are not moving through water, dramatically moving through water, to move closer to my root. And to be able to get those, I have to create a very large surface area to create that diffusion gradient. And that's why we see these extensive root systems. Now, add on to that, where in fact do they put these root systems? They're not putting them everywhere. They're very selective at where they put them. Generally, plants put an extensive root network right up at the surface of soils. Why? The answer is up there, but why? Well, they're going to do it because that's where the nutrients are. Then I'm going to ask you, and the next question is, why are the nutrients there? Why are the nutrients concentrated at the soil surface? What? That's where they, what? That's where they get added. That's where they're harvested to. Around here, every fall, the soils get fertilized. And they all get fertilized at the surface because of leaf fall. Okay? Why should a plant put roots way down deep if it's looking for nutrients if there aren't any nutrients there? It's going to put the roots where the nutrients are so it can optimize it. Now, it's not to say that they don't do deep roots, but the deep roots are generally not for nutrients. The deep roots are for water. Okay, so when you guys have dug up a plant or something like that and you look at the architecture and it looks like, something like that. Okay, it's not like a box. It's very much like this, right? That's because it's spending a lot of roots this way to get those nutrients, as well as some water, but it's sending roots down deep so that it can get water, as well as some nutrients. But most of the nutrients are at the top, most of the water is down below. Okay, does that make sense? And it's due to this issue, for nutrient-wise, it's due to this issue of both mass flow as well as diffusion. Questions? If you feel comfortable with this? All right. If we're talking about roots, we need to talk about a little bit, just one slide, we need to talk about what are in fact potential inhibitions to that root. Okay, what is it that limits those roots? And there's basically three of them. Physical, totally makes sense. I move into something that has a bulk density greater than 1.65. Do you guys remember that one? Okay, at that point, roots are inhibited. They can't necessarily penetrate. The density is too much. Okay, I also can have chemical limitations. Certainly when the systems go wet, I'm going to have oxygen deficiencies. But I also can have stunted roots based on some issues. I don't have enough calcium, or you saw the lettuce slide in the very beginning. That was aluminum toxicity. The roots aren't growing. Okay, if there's too much roots in there, in essence, we're looking at a pH control here. Low pHs, more aluminum. The aluminum is toxic to the roots. The roots can't grow, so I have root stunting. Make sense? Okay. Also, you can have biological responses, certainly something grazing on it, but often root diseases. Okay, many of these root diseases are in fact related to water, but I could have root rot. Okay? Question? Not a question? No? Okay, any questions at this point? Do you guys feel comfortable about this? Okay. So, what we're going to be doing on Wednesday is we're going to be changing the slant of this lecture. And we're going to be talking more about nutrient management and fertilizers. I still want you guys to be thinking about these 6 things. What are the constraints on this system? What are the things that are controlling the system?